Awesome. Cool. Well, thank you so much for inviting me to give a talk. My name is Erika DeBenedictus, and today I'm going to be talking about using evolution to do engineering. This is my favorite sort of visual for using evolution to do engineering, which is called directed evolution. This shows the evolution of corn. Way on the left, this weird sort of wheat-looking thing, that's teosinte, which is the great ancestor of modern-day corn, which farmers arrived at after 6,000 years of cultivating corn over many rounds of diversifying the corn genome and selecting the best plants to seed next year's crop. Farmers were able to do a remarkable amount of essentially genetic engineering on the corn genome without knowing anything about DNA. And that's the power of directed evolution. And today I'm going to talk about a directed evolution method that I created called PRANCE, which allows us to do very rapid protein engineering. And I'll talk about how I applied PRANCE to sort of address two aspects of evolution that make it really difficult to use in an engineering context, which are when things evolve, sometimes they go extinct, not so good for engineering. And also when things evolve, it's quite a random process, which is also not ideal for engineering. So one of the first difficulties with using evolution to do engineering is it takes a really long time. So in the natural world, we're surrounded by all these amazing organisms and proteins, which took 6 billion years to evolve. And even in this example, which was much faster, it still took 6,000 years to evolve corn, right? And as a PhD student working on directed evolution, I was rather hoping my PhD would take less than 6,000 years. And in this instance with corn, one of the reasons it took so long is because the life cycle of this organism is really slow. So you can only plant corn once a year, and it takes like multiple years for the plants to grow and for you to sort of see what happened. And a really good way to accelerate engineering with evolution is to use an organism that has a much faster life cycle. And so one such organism, which I worked with, is the M13 bacteriophage. So this is a virus that can infect bacteria, and it can perform its entire life cycle in 20 minutes flat. So this is an organism that has a really fast life cycle, which we can use to speed up directed evolution. And in particular, there's a technique called phage-assisted continuous evolution, which relies on the fast life cycle of the M13 bacteriophage to do super fast evolution on specific proteins. And basically the way it works is you begin by engineering some bacteria to contain a sensor that will detect the activity you're attempting to evolve in some sort of protein. So it has essentially a gene circuit in it that will detect the protein activity you want and produce a gene when the sensor triggers. The next thing you do is you take the protein you're trying to evolve and encode it on the genome of the phage, which allows you to deliver that DNA into bacterial cells in huge numbers. So you're able to evolve really large populations with this technique. Once the DNA encoding your evolving protein is inside the bacteria, it will be transcribed and translated, and then the sensor inside of the bacteria will evaluate that protein. If the protein has no activity, kind of nothing more will happen. But if the protein does have activity that you're looking for, the gene circuit will trigger. It will produce this essential phage protein called P3, and that protein will allow the virus to propagate. And what's very clever about this setup is you can create a cycle. So viruses encoding better versions of your protein will produce lots of baby bacteriophage, which will go on to infect a next set of bacteria. And indeed, in this cycle that's set up on the right side of this diagram, you have all of the essential components for directed evolution. You have selection, so you have the bacteria distinguishing between proteins that don't work and do work. And then you also have diversification. So when the phage genome replicates to produce progeny phage, that process is error prone and can introduce mutations into the gene. And so PACE altogether can be used to evolve kind of any protein for which you can engineer a gene circuit to detect that protein's function in bacteria. And this is like a small sampling of some of the gene circuits people have used. People have evolved everything from polymerase promoter recognition to protein-protein binding with sort of like a 2-hybrid approach to aminoacyl tRNA synthetase charging, which I'll talk about. Now, traditionally, sort of the equipment setup for performing a PACE experiment looks kind of like this. Basically, you have to culture bacteriophage in a bioreactor, and that involves feeding containers that contain populations of bacteriophage with fresh bacteria that has just been cultured, and then removing waste. So in this picture, there are 8 independent populations of bacteriophage that are in these sort of central small vessels, and they're being fed by bacteria that's being produced in these 2 Turkey vessels on the side. Now, fundamentally, the thing that sort of limits traditional setups in terms of how many independent experiments can be conducted at a given time is that when you use sealed flasks and you use peristaltic pumps and tubing to create a bioreactor, every bioreactor has to have 1 in tube and 1 out tube. So while I was in grad school, I decided to make what is actually a very small modification to sort of the equipment that's used to conduct this experiment, which is that rather than performing the liquid transfers with tubing and pumps, I decided to perform the same liquid transfers, but with a liquid handling robot. And so the way that works is rather than constant inflow and outflow, you have a pipetting head that comes by, adds new liquid, mixes and removes waste, and then off it goes to service some other container in the intervening time. And making this really simple change to the equipment that's used to conduct this experiment has a really big impact on how many experiments you can run. So you end up with something that looks kind of like this. You have a liquid handling robot with a 96-well pipetting head that is performing liquid transfers for 96 independent experiments at once in each of what can be several 96-well plates on the deck of the robot. So you've gone from a system that can perform sort of a handful of independent experiments at once to hundreds of independent experiments at once. And then on top of that, hiding over here in the corner is this integrated plate reader, which allows us to take readings about what's going on in each of those hundreds of populations as they evolve, which, as you'll see, is actually quite useful and actionable data. So altogether, I created this system that's called PRANCE, which stands for Phage and Robotics Assisted Near Continuous Evolution. And it allows us to perform this really rapid type of virus-assisted evolution in very high throughput with many independent experiments, and to also monitor those experiments in real time as they go along. Now, for the rest of the talk, I want to describe how I applied PRANCE to address a couple features of natural evolution that are inconvenient when you're trying to use evolution to do engineering. So namely, those are extinction. When natural things evolve, sometimes they just die, which when you're trying to engineer a protein is not useful. And also randomness. So evolution is a random process, which is not always ideal for allowing us to sort of systematically and robustly engineer things. So in a little more detail, talking about extinction first, when you have a population that is subjected to some sort of environment, if the challenge that environment poses to the population is just too hard, instead of evolving, the population will just entirely die out. And that's not beneficial if you're trying to use evolution to engineer a protein. Obviously, if everything dies, you don't get an evolved protein and your experiment has failed. And actually, there's also a converse problem, which is that if you subject your population to an environment that is too easy, also nothing will evolve. And neither of those outcomes gives you what you need, which is a protein that has some new mutations that improves its function. So sort of with extinction, we face this kind of Goldilocks situation where, we need to figure out how to create this environment that is just right just hard enough to entice the population to evolve, but not so hard that we kill it off entirely. And this can be quite challenging, and is actually made more challenging when you do sort of multiplex evolution, and perhaps are evolving many different biomolecules that begin with different amounts of initial activity. So in this experiment, I was trying to evolve some tRNAs that had been engineered to decode new codons. And the tRNAs that I initiated the evolution experiments with began those experiments with vastly different initial activity. So this arginine tRNA was like 10 times better than this phenylalanine tRNA. Those were just the starting genotypes I had on hand. And when faced with the task of trying to come up with some sort of environment that would be successful at evolving all four of these, one reasonable place to start, because they have both lots of activity and very little, would be to subject all of them to sort of a moderately difficult environment. And unfortunately, it doesn't work. So if you subject all four of these biomolecules to a moderately challenging evolution environment, 3 of them go extinct immediately because it's already too hard, and 1 of them doesn't evolve in the slightest because it's already too easy. And indeed, in order to succeed in evolving these biomolecules, in some cases, you have to make the environment more lenient, in which case you're successful at evolving the biomolecules that have very, very little initial activity. Or in other cases, you have to make the environment more challenging, in which case you can succeed in evolving the biomolecule that has the most initial activity. But altogether, it's obviously very difficult to sort of get the environment right. And so in order to create a systematic solution to this problem, I decided to implement a feedback controller. So how this worked was I handed control of the environment over to the robot, and I tasked the robot with customizing how challenging the environment was in response to measurements of how every single population is performing individually. So every population receives a customized environment that is designed to be the Goldilocks environment for that particular population. And this feedback controller was 100% successful at getting these biomolecules to evolve, despite the fact that they have really different initial activities. So the way this worked was I told the robot to switch each population over from easy to medium to hard, from red to blue to green, as soon as it saw that the population was reasonably sized and performing rather well. For phenylalanine, which was the biomolecule that had the least initial activity, it stays on easy for a real long time. It stays on easy for nearly 20 hours before it starts being subjected to a more challenging environment. In contrast, for arginine, which was the best performer initially, it hardly is in the easy environment at all. It gets bumped up right away. And the other 2 biomolecules experienced, again, customized environments for their situation. And so altogether, we can solve this extinction problem by implementing just a good old feedback controller, which can be used to hit that Goldilocks level of difficulty for every population individually, and thereby make engineering with evolution a more robust and predictable process. Now, the second category of sort of undesirable features of natural evolution that we have to deal with in engineering is randomness. So evolution is a stochastic process. Evolution happens when random mutations accumulate in genes that alter their function. And when you use evolution to do engineering, you can get to the end of a evolution campaign with kind of like existential questions of like, okay, here's my protein, it has some mutations in it, it clearly has higher activity, but really what I want to know is which of these mutations mattered. Are some of these mutations just random? Am I seeing them just by chance? Or are some of these mutations actually functional and contributing to the altered function of this protein? And I decided to address the randomness of evolution by doing better sampling. So in this experiment, I was evolving the T7 RNA polymerase to initiate transcription on a new promoter. And rather than perform this evolution in one population or a couple populations, I decided to do this experiment in 96 plex. So there's 96 independent populations of this polymerase that are all competing to find solutions to this evolution puzzle, which is how do you initiate transcription on this new promoter. And as you can see from the real-time monitoring, all 96 populations are successful at finding an evolved variant, and those evolved variants start overtaking the population sort of between like 18 and sort of like 26 hours. It's sort of in that time range. And when we got to the end of this experiment, we picked 12 different populations, and we sequenced a bunch of individual polymerases that we found within those populations. And what's really cool about doing these multiplexed evolutions is it becomes really easy to tell actually which mutations matter, right? So in this experiment, it's really obvious that mutations at residue 219 and at residue 748 are the mutations that are altering how functional the polymerase is and thus driving fixation of those genotypes in the population. And we can tell that because they're convergent amongst many independent experiments. Whereas there's many other mutations that we see, but they're not convergent. And so we can infer that those are not the ones that are functional. And as an additional little bonus, this was not something we were looking for, but this was very cool to see. We actually noticed that if you look at the kinetic data, we have on hand knowledge of exactly when each of these populations evolved. And as you saw on the previous slide, they had some sort of a time range of sort of between 18 and 26 hours or so. And when we looked at the data, it sort of seemed to be the case that how fast the populations evolved seemed to be related to what genotype we found in the populations at the end. And you can see that just by eye, but I mean, it is also statistically significant. And this was like a bit of a head scratcher for a while. But eventually we figured out that actually what's going on under the hood is that the mutation at 219 is a transversion mutation, while the mutation at 748 is a transition mutation. And based on the mutational spectra in these bacteria, the transition mutations are way more common. And that's why the populations that generally evolve earlier are ones in which we find transition mutations, and vice versa. Later evolvers tend to be the transversion mutations. So anyway, this was like a sort of cool tidbit of how sort of having this combination of mutational and kinetic data allows us to draw further insight into sort of why we see the genotypes that we do. And altogether, we're able to understand which mutations matter by comprehensively sampling many populations, attempting the same evolution, and using that to infer which mutations are functional. And as one final, very brief example, there's sort of a related question with randomness, which is what do the mutations do? How are they altering the function of the protein? In this experiment, we were evolving some aminoacyl tRNA synthetases to charge tRNAs with non-canonical amino acids. And we performed this evolution in quadruplicate. And in addition to doing it in quadruplicate, we also did it under two conditions, one in which we added the non-canonical amino acid, and the other condition in which we did not add the non-canonical amino acid to the media. So we have this no non-canonical amino acid condition, which acts essentially as a negative control. So if we saw evolution occur under that condition, we could infer that the genotype that we initiated the experiment with is prone to evolving promiscuous charging with canonical amino acids. And because we didn't see any evolution under that condition, we concluded that what was most likely was that the mutations acquired over here in sort of the positive control serve to improve integration of the non-canonical amino acid, not a canonical one. And indeed, when we checked with a separate assay, that was confirmed. So essentially, by conducting what I'll call evolution controls, we can sort of infer more about what mutations do that we see in these experiments. Now, all of this work was done in the lab of Kevin Esvelt, who was my advisor in graduate school. Emma and Brian both worked sort of more on the molecular biology side, and Dana and Stefan both did a lot of the sort of software engineering that was necessary to get all of this to sort of work together. And you can read more about this in both of these papers. The one on the bottom is specifically about PRANCE, and the one above it is about some other stuff we did with this automation. And before I close, I just want to mention a program that I started recently that I'm really excited about. So a question I get a lot when I talk about PRANCE is this question of access, right? So we have the technology to make liquid handling robots, but not everyone has access to it. And indeed, I think a big reason for that is that as a person whose lab owned a liquid handling robot in graduate school, I think the sort of current paradigm for how we access these sophisticated instruments doesn't work for very many people. No one wants to buy a robot. They're expensive. The service contracts are expensive. It takes ages to train people in how to use them. And by the time they're trained, they graduate. And sort of altogether, this system doesn't work very well for most people. And there are great alternatives like having these instruments be housed in sort of core facilities or biofabs or cloud labs. And as an effort to sort of make automation more accessible to people, I recently started this program called the Bioautomation Challenge, which is a grant program that you can apply to. Deadline is March 16th. If you have a method that you would like to automate, you can propose that method, and we will be selecting proposals, and those groups will receive a year of access to a cloud lab facility as well as a reagent budget. So it's my hope that sort of by changing the paradigm with which we access these instruments, we can better integrate automation into science, making it more reproducible, scalable, and easier to share and sort of replicate. A final thank you to my advisors. I've benefited from a very interdisciplinary education, and it is thanks to these people as well as too many colleagues to count. And happy to take questions.
Thank you very much, Erika. I just realized I forgot to, oh, there's a lot of hands clapping. I forgot to introduce you at the beginning. So Erika, Dr. Erika Benedictus has gained her PhD work. So you talked about your PhD work mostly today, weren't you? Yes, yeah. At the MIT in bioengineering. And since then, that she's been doing protein designing at University of Washington in Seattle in David Baker's group, and she is in the process to hop across the pond and move to the Greater London area, I hear. And so, welcome to the Big Smoke. All right, so. The questions, there's one in the chat, and if you have questions, please raise your hands. So Andres, would you like to ask this yourself? If so, you can turn on your mic, but if you prefer that I lead, I can. So Andres is wondering, how would you avoid the bacterial defense when the phage infection becomes a cycle?
Yeah, so the way it works in this system is that you, so in the sort of equipment setup, you are constantly feeding the bioreactors with fresh bacteria that have like never seen a phage before. So every bacteria is sort of like single use only, and the phage infect, and then produce progeny, and then the bacteria, they all get sort of, they get washed out, so they get, you know, bleached. So what's nice about this is you kind of don't need to worry about anything evolving in the bacterial genome because every time you use a bacteria, it's something you've prepped freshly. And of course, we just use bacteria that sort of don't already have CRISPR in it, or something like that.
Thank you. Does that answer your question, Andres? Yes, thank you, he says. Any other question? I am wondering about the automation challenge. When you say we for that program, who are they? So you and?
Yeah, so this was an idea that I came up with, and I pitched it to Schmidt Futures, who's funding it. And then we're partnering with Emerald Cloud Lab to provide the Cloud Lab facility. So we is me, Schmidt Futures, and Emerald.
I see. Yes. Thank you. And the deadline for our idea is, the first round is?
Yeah, it's March 16th. It's a pretty minimal application. You submit sort of a description of the project you want to do, a protocol, and then there's some sort of short answer questions. So it's not tremendously onerous. And yeah, I think there's lots of interest in it. So I've been talking especially to lots of people who are sort of in the protein engineering community. And it's a cool opportunity to sort of develop, especially measurement techniques for if you want to characterize proteins. So it's a really nice way to collect like sort of larger, higher fidelity data sets than is otherwise easy. But all sorts of proposals are welcome.
Fantastic. Did you say like that anybody else who are eligible? I guess, does it have to be?
Yeah. So yeah. It doesn't have to be. So one of the cool things about the Cloud Lab is like, we can give access to anyone who can like log into the software on a computer, right? So we're accepting proposals primarily from academic labs, but also from like community bio spaces, or also like companies, like startup companies especially, are also welcome to apply if they have ideas.
Fantastic, thank you. Yeah, that sounds really cool. Karen, would you like to ask question yourself?
I can. Okay, I can. I finally got my microphone back. It had a little. So thanks a lot. It was a really great talk. And I really like seeing the fact that evolution is starting to be employed in synthetic biology, because I think in the beginning it was very much we must be rational and we must try to fight against evolution. So I was wondering what you think are the prospects going forward for using evolution in synthetic biology, and will it become routine?
Oh, I mean, this is a great question. I mean, I'm currently a postdoc in the Baker Lab, where you're not allowed to so much do directed evolution, because the whole idea is we want to learn how to do these things rationally, right? And I think, I mean, I guess my opinion is it's all about bootstrapping, right? So I'm happy to use evolution to engineer things as long as it gets me closer to a universe in which I understand how those things actually work and maybe can make them de novo, whatever that means, right? And I think, yeah, I think there's maybe going to be a marriage of like sort of this very bottom-up synthetic biology and kind of the more, open to evolution as a solution community. So yeah, I mean, I think the way forward is like do a little of both and not be quite so principled about it.
Great, thanks a lot.
Thank you. Is there any other question? Yes, Son, your hands up. If you want to ask a question, please go ahead.
First, thank you very much for your fantastic presentation. And please excuse my lack of expertise for the liquid handling system. But as far as I know and understand, I think for the evolution process, the evolution pressure for each different protein targets are very distinctive. So how can you adapt your whole system for different need for the evolution of different proteins, for example?
Yeah, so I mean, in this particular instance, the way you specify what the activity is that you're trying to evolve is not on the automation side at all. It's through engineering a gene circuit inside of bacteria. And so that process itself has traditionally been rather onerous. And I think there's lots of ways to imagine sort of accelerating the process of how do you create a gene circuit and tune it and all of that using automation. So I mean, I think that's like a big area that we could just be better at, making gene circuits. But yeah, I would say also, like, on the automation side, you can to some extent tune the stringency, so how strongly, how challenging the environment is, with very simple techniques. Like, you can change how fast the flow-through rate is through the bioreactor. So if you make it faster, the bacteria, they have to replicate faster and be fitter in order to survive, right? So there's sort of like general purpose ways also of adjusting the experiment that are entirely on the robotic side as well.
Great, thank you. So we do have a more question popping up in the chat box, but we have another talk, a great talk today. And so what happens is the speakers, both of them, graciously agreed to stay on the talk, on this call. And so if you have more questions, they can answer. So would that be okay, Keisin and Stoyan? If you can stick around, then we can have more Q&A with Erika then.
Yeah, sure. Thanks.
Thank you very much. Thanks, Erika. Thank you very much. And see you later. So the next speaker is Mateo Sanchez Lopez. Is currently postdoc in the Center of Synthetic Biology at Imperial. He has, well, actually, maybe, yeah, well, actually, I don't know.
Okay. Okay.
Yes, if you can introduce yourself.
No worries. I will be happy to introduce myself.
Yes.
Well, so I did my PhD in organic chemistry in Spain, and then I moved with Alice team to MIT and later in Stanford to work in neuroscience, protein engineering, and directed evolution. And then, when I finished my project, and COVID happened, so I couldn't apply for a lot of jobs, and I decided to come to England. And I did, I'm doing my second postdoc with Tom Ellis in synthetic biology and artificial chromosomes. Yeah?
Thank you. Perfect.
Very welcome. Okay, so today I'm going to present like the work of my previous work about directed evolution and protein engineering. Can you see all of my screen?
Yes.
Okay, wonderful. So the title of the talk is Mapping and Manipulation of Neuronal Circuits by the Directed Evolution of the TEF protease. One of the biggest challenges of this century for humankind is to understand how the brain works, especially in terms of single-cell resolution. So the brain has billions of neurons. However, only around 100 to 100,000 of these neurons are involved in any specific behavior. For example, if we give electrical shock to mice, or if we provide chocolate milk, the neurons that are going to be responsive to these stimulus are very different. So a big challenge is how to identify those neurons. Well, and we have some tools that to measure and to manipulate neural activity. For example, we can measure in real time neuronal activity by using fluorescent proteins that are called GCAMs. Okay, and the advantage of GCAMs is that circular permutated GFP, that in the presence of calcium, that is a good proxy of neural activity, they are going to restore the fluorescent signal. However, if you use a microscope, you can only target a small field of view. It doesn't allow further correlation of the study, like to replace the behavior of interest. Then, if you want to replay the behavior of interest and you want to further manipulate that neuron, like by silencing or activating it, you can use opsins. Opsins are transmembrane proteins that, in the presence of light, they process a downstream signal effects that they can silence or activate the neurons. So you can establish a causality, but the problem is that you don't know previously which neuron do you have to target, okay? So the ideal tool to identify, to map, discover, and manipulate the active neurons or the active neural circuits underlying a specific behavior is a tool that can work in a large brain, in a large region of the brain, because the neural circuits are very scattered. And also you can mark the neuron, you can label the neuron with a fluorescent protein to do imaging and to discover which neuron has been activated, and then an opsin. Why? Because you want to activate this this subset of neurons in order to establish a causal relationship. So what we want is to link neuronal activity with gene expression, and we want to do that in a specific temporal window, okay? So there are some tools that do that. For example, a pioneer example was developed by Mark Myford in Science 2007, where he uses immediate early genes in the presence of a drug to have gene expression. So what it does is, or what they did, is to place a transcription factor under the promoter of immediate early genes. So these genes, every time that there is neural activity, they are going to promote the transcription of this druggable transcription factor. And in the presence of a drug that it matches the behavior of interest, this transcription factor is going to recognize its promoter and is going to promote the expression of the reporter gene. So in this way, we can have gene expression as an output, so we can further manipulate the circuit. But the problem is that these immediate genes, they are not very well characterized. They are not universal in any organism, and the temporal resolution can be from 2 hours to 6 hours. So this is very far away of the temporal scale that neurons uses, which is normally in the time scale of seconds to milliseconds. In 2015, Marcus Reiter in Janelia, they developed a tool called Campari that they use calcium and violet light. And calcium is, as I mentioned before, is very important because it's a universal marker. And now the output is a change in color. So here, what they have is a GFP, a circular permutated GFP with calcium-sensitive modules that, in the presence of high calcium and ultraviolet light, they are going to promote a photochemical reaction that is going to change the color in the chromophore, switching from a GFP to RFP. So here, the advantage of this tool is that now it's responsive to calcium, which is a good proxy of neuronal activity, and it has an excellent temporal resolution, which is in seconds, okay? Because this is a photochemical reaction. But the problem is that the output is a change in color. So what we want is to combine the advantages of both tools. We want to have calcium and light as an input, and then we want to have gene expression as an output, okay? We want a tool that works very fast and it has a transcriptional readout. So the Team Lab in 2017 developed one tool that is called, the acronym is FLAIR, for fast, light, and activity regulated expression, that where we have light and calcium, and we have reported expression as a readout of the tool, okay? So we're going to release a transcription factor here that in a specific time window. So we're going to take a snapshot of the neuronal activity in a specific time window, and then we are going to replay the behavior of interest. And one question is, how do we build such a tool? Well, so here what we have is our orthogonal transcription factor, for example, TTA, that is going to be anchored to the plasma membrane. Therefore, is incapable to go to the nucleus and to do its job, and we're going to link it to a protease cleavable site, okay? This protease cleavable site is going to be caged by a LOB domain. LOB domain are small proteins from plants that are sensitive to blue light. Later, we incorporate the calcium sensitivity, but in the fusing a calmodulin to the TEF protease, later I will say why, and then we have here the MK peptide, okay? So every time that a neuron fires, there is going to be an increase in calcium concentration, and when this calmodulin sense this calcium is going to suffer a conformational change, and now this new form of calmodulin, it has a huge affinity against the MK peptide, that is the calmodulin binding peptide, and it's going to bring in proximity the TEF protease to the TEF cleavable site. However, nothing can happen because in dark conditions, the LUV domain is protecting from the cleavage this recognition site. However, when we shine blue light, there is going to suffer a conformational change, and now the protease can cleave its peptide substrate, and now the transcription factor is capable to be translocated to the nucleus and to promote the expression of the reporter gene. So here you can see a scheme where deciphers how this tool works, and it works like an AND gate. So we will only see expression of the reporter gene, in this case M cherry, when we have light and high calcium conditions. So after developing this tool, we tested in neuron cultures. This is neurons from mice or from a rat. And here you can see the expression of the tool doing anti-V5 expression. And now here in the right, you can see that only when we have high calcium concentrations induced by media change and only under light illumination after 10 minutes, we can see a very nice expression of the M cherry. Okay, and the signal to noise ratios are very good. This was a breakthrough in the field because we decreased now the time that we use to label active neurons from 30 minutes to, from 6 hours, 2 hours to 10 minutes. However, it's still far away of the time scale that neurons use to process information that happens in the time scale from milliseconds to seconds. One of the goals in my postdocs was to improve the sensitivity of this tool. And if we pay attention to the design, the rate limiting step here is the amount of transcription factor, the molecules that are released from the plasma membrane when we have an increase in calcium concentration. And it's worthy to say that this increase in calcium is very transient in neurons. So the TEF protease is excellent protease, but still is a little bit slow because the K-cut is around 0.2, and only 2 molecules are going, 0.2 molecules are going to be released, or cuts are going to happen per second. So if we want to increase the sensitivity, what they have to do is to improve the velocity of this enzyme to reduce the tagging window. So then what I did is to use directed evolution to improve the catalytic efficiency of the TEF protease. And I know that this talk has been a lot of protein engineering, but I found this field fascinating. And now let me explain why TEF protease. Okay, so TEF protease has very specific sequence, okay, that is orthogonal to the mammalian proteome, and that makes it ideal for cellular biology and biomolecular tools. Also is monomeric, stable, and very easy to handle. So for all these reasons, has become the protease of choice from biotechnology and for a lot of biomedical applications. Then directed evolution, as Erika said before, is a method in protein engineering where you use you introduce random mutations, and then you have a selection vector that is going to select for the physicochemical properties of the enzyme or protein of interest of what you want. And looking in literature, there are 2 papers that were published where they used directed evolution and they applied to the TEF protease. One was published by Iverson in PNAS in 2013, and the other was used in PACE, that Erika is one of the techniques that, well, the first technique, and then come PRANCE, by David Dewlap in 2017. But these platforms are excellent to change the selectivity of the TEF protease, the substrate specificity, but we don't want that. What we want to is improve the kinetics. Later will be very nice to discuss why I couldn't apply PACE to my selection. So so I couldn't work with this platform. So what I did is I built a new platform for directed evolution of faster proteases, and in this case, the TEF protease. And I do that using yeast for several reasons. We have a transcription factor anchored to the plasma membrane in the same design as FLAIR, that through the TEF cleavable site, which is caged by this L domain. So the only difference is now we have the CRY and CyBM, and these 2 proteins come from plants that are sensitive to blue light. So they are photo-induced dimerizing pairs. And now here we have the TEF protease, which is also fused to the M cherry just as a protein expression marker. So in the presence of blue light, the CRY is going to suffer a conformational change, increasing its affinity against CyBM, And now it's going to bring the protease close to the recognition site. The LOB domain is going to be open, making accessible to the protease. And now the transcription factor can be released from the plasma membrane to the nucleus, promoting the expression of the reporter gene, in this case, GFP. And this platform is ideal because it works with low affinity proteases, because we require a protein-protein interaction for this cleavage to happen. Here you can see in this FACS plot that we have this MCHR intensity, which is going to measure the TEF protease expression, and only with the presence of light we can see an increase in the expression of the transcription factor. In the reporter gene. This GFP intensity is going to reflect the quantity of transcription factor that has been released, and this is another way to measure the activity of the TEF protease. So you can see how the GFP expression scales with the radiation time. So then what I did is I use directed evolution. So I generate a library of 10 to the 8 variants using error-prone PCR. This is one of the disadvantages of these methods compared to PRANCE or with PACE, that the pool is fixed. And then if you want to introduce more mutants, you have to do more rounds of error-prone PCR, but in this case, it's okay. So I generate 10 to the 8 mutants, and then I use homologous recombination. That is a process that is very good or very efficient in yeast, to make my TEF protease library. Then I'm going to shine light, and I'm going to culture in dark conditions the yeast culture to allow the GFP transcription and translation. And I'm always going to harvest the brightest yeast. And my selection scheme that I'm going to apply here is to decrease the amount of time that I shine blue light. And why? Well, because if the entire pool of mutants, if the entire yeast population is illuminated with the same amount of light, now the faster proteases are going, in the same unit of time, they are going to release more units of transcription factor, and they are going to increase the expression of the reporter gene. So I'm always going to harvest the brightest yeast, and I'm going to decrease the amount of time that I shine blue light. Okay, so this is how I increase my stringency to select for the faster proteases. Here you can see that this is the wild-type TEF protease that requires 30 minutes, okay, to saturate the signal in the reporter gene expression, but in 13 seconds nothing happened. After then, I introduce the mutations by homologous recombination, and some of the mutants are dead, and that makes total sense because, for sure, there are some mutations that are impairing the catalytic activity. After one round of sorting, I see that after 30 seconds of illumination, there are some mutants that show some activity. But then, if we keep doing fast sorting of 3 rounds of sorting, now we can see a population that saturates the signal. And now we can compare it with the wild type that in nature, what before it takes 30 minutes, now we have saturated signal in just 30 seconds, which is pretty amazing. When I analyze the individual clones, this is the crystal structure of the TEF protease. And here in blue, you can see the ligand, the peptide substrate, and in red, this is the catalytic triad of the TEF protease. So when I analyze several clones, what I observe is that most part of the mutations were surrounding the catalytic triad. And that makes sense, is agree with the data, because here, what we are doing is improving the K-cut, okay? We are increasing the activity, and we are not switching the affinity or the selectivity against the peptide substrate. So this is why these mutations are surrounding the catalytic triad. Later, I did a side-by-side comparison of my favorite mutants. One of them has just one single mutation that has an incredible effect, and the other is a double twin mutant that I call UltraTEF1 and UltraTEF2. And when I compare them with the original TEF protease, you can see that in 30 seconds, the signal is very high. The signal that is measuring the quantity of transcription factor that has been released, that is an indirect way to measure the activity, and in 5 minutes still is much better. Then I did some kinetic analysis of the winning clones, and here you can see that I have a construct. This is the maltose bundle protein fused to the GFP through the TEF cleavable site, And then here, I'm incubating with the different TEF protease at different times in the absence of presence of the TEF protease. So here, if there is no TEF protease, there is no signal of the GFP. But with increasing times, you can see that there is a much higher increase of the appearance this band that matches the EGFP that has been released, that has been processed for this construct. Here you can see some kinetic analysis, and you can see that it's improvement in the K-cut around 5 to 6 fold with these new mutants. So if you remember, this is just to measure this process was to increase the K-cut. But now if you ask, well, and what happened with the affinity? You don't care because the TEF protease is so used in biotechnology for proteomics analysis to remove affinity tags that also will be useful to have a TEF protease that is very active and with a high affinity. So in this case, I performed the same experiment, but now I remove the CRISE IBM. So I'm going to select for mutants that instead of having a K-cut, they have a low Km, that the biophysical meaning of that is to have a high affinity. So here, what I'm going to select is for the proteases that has high affinities to the TEF cleavable site, and I'm going to sort the brightest gist. Here you can see a comparison between the wild-type TEF protease and the one that has my favorite mutation, and there is a huge increase, but I'm going to use this as starting template. Again, after 3 rounds of directed evolution, you can see a huge increase in the signal that is related with the release of the transcription factor. And here you can see again some analysis, some kinetic analysis, where now we have a variant that I call Ultra TEF3 because it has 3 mutations that has a Km 3-fold less and a higher K-cut around also 3.5 times. So this was great because we were able to evolve TEF protease with different activities and affinities. But now what we wanted to do is to increase the affinity in this process when the TEF protease is in proximity to the TEF cleavable site. So here we have a side comparison of FLAIR that was the first paper published in Nature Biotech in 2017. Here you can see the expression of the tool in neurons from rat. This is a rat culture. We've used the GFP to avoid immunostaining. This is the expression marker. And now you can see that we will have field stimulation that is a milder, much milder way to stimulate neurons compared to media change. And if we shine light for 5 minutes, the expression of Zen cherry is very little. However, when we introduce these magic mutations, now you can see that the expression of Zen cherry is much higher, and now we were able to label the neural activity for in a time scale of seconds for the first time. Also, I would like to highlight that this tool really works like a AND gate, and the best way to measure that is to stagger the behaviors. So this is telling me that to measure that we are measuring the neural activity just in a specific time window. So for example, if we first shine light, blue light, we keep in dark for 1 minute, and then we stimulate to induce an increase in calcium concentration, or if we do the opposite, that first we stimulate the neurons in dark conditions, and we wait for 1 minute, and then we shine light for 5 minutes, the expression of Zen cherry is very little. So this is the proof that this tool works like an AND gate. So we are measuring neural activity in a specific time window. So then, in collaboration with Professor Christina Kim, that she was a postdoc in the lab at my time, and with Professor Carl Daysroth in Stanford, that he's one of the fathers of optogenetics, we use this technology to make real discoveries, biological discoveries, and for example, we were interested in knowing which neurons in the brain are responsive to nicotine. So to do that, what I did is I make adenoviruses that encode the FLAIR viruses, and which we inject them into the nucleus accumbens of in the brain. Then we wait 5 days for the expression of these viruses, and we inject in the intraperitoneal some nicotine. We shine sunlight while we were doing the injection of nicotine, and then we wait 1 day for the expression of the reporter gene. And here, we were happy to see that we discovered some neurons that were stimulated by the nicotine. So as the turn on of the tool is expression of a reporter gene, we were able to isolate these neurons using FACS, and now and then what we did is to molecular characterize the neurons that are responsive to nicotine, which it has a very interesting biological value. Also, another big question in biology is to understand how the neurons are connected and to know what is the connectivity, the connectome of the active neurons. So to solve that problem, what we did is to combine FLAIR with optogenetics. So the question here is to know which neurons are connected from region A to region B. So if we use the FLAIR technology, we can solve that. What we're going to do is to use excitatory opsins, and as I said in the introduction, opsins are transmembrane proteins that they can activate or silence neurons. So here we have in the glutamatergic neurons in the prefrontal cortex of the brain, we are going to express this channel rhodopsin that is sensitive to yellow light, therefore is invisible to FLAIR, because FLAIR is sensitive to blue light for the LOB domain. And now in the other part of the brain, we are going to inject the FLAIR viruses, okay? So when the neurons are synaptically connected, these neurons here, we shine blue light, we shine orange light in this part of the brain, and we are going to record the neuronal activity in the nucleus accumbens with blue light. So the neurons that are synaptically connected, they are going to excite these neurons, and they are going to elicit an increase in calcium concentration. So here we have a transgenic mice that express channel rhodopsin in the prefrontal cortex, and now we inject the viruses, and here you can see that there is an effective communication between the prefrontal cortex and the nucleus accumbens, because we only were able to see expression of den-cherry in the animals that have this channel rhodopsin. And this is very important because now, again, we were able to do single-cell analysis and RNA-seq and other omics techniques to characterize these neurons, and also playing the opsin, we solved that the behavior of the neurons they drive an adverse behavior. So we were able to restore a phenotypic effect. Okay. So all this technology is public available, so that's the thing that made me happy, that a lot of people are using it, and for sure, you know, if you want any of the constructs that I disclose here, they're uploaded in Argin, so it's free to use. And finally, I would like to send my previous advisor, Alice Ting, that was brave enough to hire an organic chemist to do all this project in protein engineering, directed evolution neuroscience. And now, you know, my actual PI, that is Professor Tom Ellis, also to, well, right now I'm working on a very different thing that has to do with synthetic biology and genomic engineering. And I would like to say thanks to my group in Stanford, where the weather is much better than in London. But yeah, and thank you to all for your attention, and I will be more than happy to take any question.
Thank you so much, Mateo. Really, really cool. It has everything, doesn't it? Neuroscience, directed evolution, and protein, you know, sensor engineering. So any questions? You can have hands up. Yes, I see. Yes, Son, you have your hands up. Go ahead and ask.
Thank you very much for your very interesting talk, and it's basically very completed. But I just want to confirm a very minor point about the directed evolved TEV, about the background proteolysis activity of that protease. You also showed that it has a very high turnover rate as well as a very high binding activity. But what about the protease activity and the binding activity of that evolved protease toward the near cognate recognition site? Because, like, theoretically, I wouldn't want a free running protease in the cell, right? And what about the phenotype of the cells carrying that evolved one?
Excellent question. So the why, that question is related with why do I choose the TEF protease? And the TEF protease is excellent because it's orthogonal to the mammalian proteome. So, for example, you can highly express in the brain of mice, and there is no effect. For example, you can express it for 1 month, and there is no difference in the behavior or any effect between the mice that express the TEF protease or the guys that doesn't express anything, because there is no protein in the mammalian proteome that has the specific sequence of the TEF recognition site. Therefore, you can use it like almost for free because there is no target, no biological target in the mammalian cell. Okay, that's one question. Is orthogonality. Second question is about the activity. Well, background is a very important issue in this when you use these tools, because when you have a transcription factor fused to the TEF recognition site and you overexpress the TEF protease, of course you can have problems with leaking. Okay, so then you have to use a low affinity TEF protease, but it's very important to highlight that now you have a LOB domain that is one protein that is caging the TEF recognition site. So you only make accessible to the TEF protease to recognize the peptide substrate only in the presence of light. And the light is the input that you use to link the recording of neuronal activity with the behavior of interest. So for example, when you express the viruses, the mice, they are 5 days, okay, and you have the electrode, but it's closed in the head. And you only shine light with a LED when you are injecting the nicotine. So this only, you are only making available for the TEF protease to recognize the cleavable site just for 5 minutes when the nicotine is present. Then the rest of the time, the LOB domain is protecting the TEF recognition site. And that's the way to avoid leakiness. Was clear my answer?
Yes, thank you very much. But yeah, I'm sorry, but maybe there are a lot more questions. But if it's possible, I guess I also want, because one of the advantage of the TEV original one is very low background proteolysis, or none at all. But because you evolved it, so I wonder, what about your evolved version? Is it like...
Wonderful question. What I did to prove that I only evolved for KCAT, but maintaining a low KM, okay? What I did is to suppress the Krebs-IBM interaction at the background was similar to the evolved TEF. So here, maybe I don't have this data here, I have it, but the best way to convince you is with this slide, where you can see that if the background of the TEF protease, if you have a lot high affinity, okay, you will see tons of background here, and still you improve the KCAT, but without affecting a lot the KM. Of course, KCAT and KM, they hold hands, okay, they are similar, they measure different things, but it's very difficult to evolve the KCAT without affecting the KM. But I think this is why the magic of this work, because you are using a protein-protein interaction to try to evolve just the KCAT, not the KM. Yes. So if you have a lot of KM, you will see tons of background here, and you don't see that. There are some, but maybe this is high background activity of neural activation, because I didn't silence these neurons.
Yes, thank you very much. Very beautiful work.
Thank you.
Thank you. Is there any other question? I am curious about your take on Karen's question before actually she asked Erika that how directed evolution and the design-led approach, how they are gonna, sort of interact, merge, how sort of into the future of a synthetic biology. Do you have any thoughts, opinions about?
Well, I'm in love with directed. Well, for me, directed evolution is part of synthetic biology. I don't see it as a different thing. For me, are like two faces of the same coin. This, and I moved with Tom because now I wanted to learn more synthetic biology, but harnessing the power of homologous recombination in yeast is one of the principles in synthetic biology. So for me, two faces of the same coin. I don't see a difference. Then if you ask me about rational design and directed evolution, I'm much more fan of directed evolution because for me it's difficult to imagine that a prediction can improve the K-cut. How to improve the K-cut? Because, for example, the mutation that I found was 10 angstroms separated from the catalytic triad. And I don't believe that by computational methods nowadays can solve that. And also, for example, you can predict a structure, but then it's insoluble, or then it's unstable, and directed evolution, you are evolving in the system that later you want to use. In this case, I use yeast, and I wanted to use in neurons, so there can be issues, but it's much closer yeast to neurons than in silico to neurons, in this case. But it would be very nice to have some computational methods to help in directed evolution. So again, they merge, but I'm not a computational guy.
Perhaps Erika can give a bit more insights into that as well. But yeah, so I guess we don't know enough about the protein sort of forms and function to completely directly sort of design. Sorry, yeah, that designed it, rationally design yet maybe. So, but okay, so I think I would like to stop the recording now, and if you have more questions, you can stick around, and you are welcome to turn on the camera and ask questions to the speakers. I will officially close the session now. Thank you so much for wonderful talks.